thank you for the very kind introduction. It's an absolute pleasure to be able to give this lecture this evening. It's also a very humbling experience, particularly when you look at the list of previous Fleming Prize lecturers. And actually, this is as much recognition of people in the lab, both past and present, and so I would like to say a big thank you from all of them, as well as from myself. So today I'm going to be talking to you about the hidden complexities of bacterial chromosomes. And the take-home message is that actually the textbook view of bacterial chromosomes and the genes they contain and the way that those genes are regulated is no longer able to explain all of our experimental observations. And what I'm going to try and convince you is that to explain all of our most recent observations, we need to go beyond our current models for genomes and their annotation and start to plunder these hidden complexities of bacterial DNA. So my talk is going to be approximately split into three parts. In the first part of the talk, I want to consider where our current view of bacterial genomes and genome annotation has come from. And then I want to consider some of the limitations associated with our current view of the genome. And what I'm going to show you is that we can no longer explain everything that we're seeing, and in particular I'm going to focus on unusual transcriptional events. Okay, and you're going to see that these unusual transcriptional events can occur in all sorts of places across bacterial chromosomes, and I'm going to refer to these as spurious transcriptional events. And by this I mean the formation of transcripts that are non-coding, have got no apparent job in the cell, and appear to have been formed almost by accident. And in the second part of the talk, I'm going to show you that parts of bacterial genomes that have got an elevated AT content are particularly prone to these spurious transcriptional events. And as many of you will know, these AT-rich parts of bacterial genomes tend to be the parts of the genome that have been acquired horizontally. So I'm talking about huge chunks of DNA containing many genes that have got an elevated AT content. These are often horizontally acquired, and what I'm going to try and convince you of is that these sections of DNA are very prone to these spurious transcriptional events. Obviously, this has got implications for genome evolution, and I'll get to that at the end of the talk. Okay, so where does our current view of bacterial genomes and the genes that they contain come from? Well, I would like to suggest that it's largely rooted in the Operon model, of course, the famous work of Jacob and Monod, and their famous paper in JMB in 1961. Okay, and they obviously won the Nobel Prize for this work a few years later. And the Operon model has clearly been very influential since it was first suggested back in the 60s. Many groups around the world have gone on to study bacterial operons and the various ways in which they're controlled. And what this work tells us is the following. It tells us that bacterial operons are groups of genes organized as a transcription unit, and these genes are transcribed to form an mRNA, okay, a polycistronic mRNA that can be translated to produce several different proteins. And of course, this mRNA is very well defined. It maps to a specific strand of the DNA. It's got a clear start point. It's got a clear stop point. And this clear delineation of the RNA is dependent on two genetic signals. At the start, we have a sequence called a promoter that recruits RNA polymerase to the DNA and tells it to initiate transcription. And at the end, we have a sequence called a terminator that tells RNA polymerase to stop. Now, clearly, this can explain many of our experimental observations and has been hugely important in helping us to understand bacterial DNA. But what I would like to argue is that actually the Operon model has become so influential that it started to blinker and bias the way in which we think about bacterial genes. And nowhere is this more apparent than when you look at the original annotation of the E. coli genome. Okay, so the E. coli genome, the sequence of the K12 genome, was published in the mid-1990s. And back in the mid-1990s, even the genome sequence of a humble bacterium was sufficient to generate a lot of interest, a high-profile publication. And very often with these genome papers that were published in the 90s, you got the opportunity to produce these kind of cartoon pullouts showing the way in which the genome had been annotated. And you can very clearly see the influence of the Operon model at play when you look at the genome annotation and you look at some of the assumptions that were made in order to annotate the genome. Okay, so clearly lots of this annotation isn't based on experimental evidence, it's based on the DNA sequence. So you look through the DNA sequence, you identify open reading frames, and then you group those open reading frames into transcription units. Okay. In addition, in this particular paper, they looked through the DNA sequence and identified sequences that look like promoters and the binding sites for transcription factors, and they used those predictions to help organize these genes into operons. Now, it's quite interesting when you look at the small print in this paper, because when they search the sequence for sequences that look like promoters and transcription factor binding sites, they included something that they refer to as the proximity factor. And basically this meant when they identified promoter-like sequences and transcription factor binding sites close to the start of a gene, those were much more likely to make it into the final annotation than promoter-like sequences that perhaps occur inside a gene. Okay, so you end up with this very simplistic view of genome annotation that's heavily biased by the Operon model. And as I've said already several times, this view of genome organization, although it can explain many of our observations, it can no longer explain everything. So what sorts of observations am I talking about? Okay, so I'm talking about papers like these, okay, and there are papers like this appearing every single week for all sorts of different bacteria and actually all sorts of organisms in the 3 domains of life. So, for example, we've got this paper up here from Joe Wade's lab, a widespread antisense transcription in E. coli. So this paper shows us that actually many genes in E. coli contain internal promoters that can drive the formation of an antisense transcript. Clearly, that doesn't fit very well with the way that the genome's annotated and the Operon view of genes and their control. A paper here from our lab where we looked at the global DNA binding properties of a transcription factor that's called RUT-R. And this transcription factor actually has the majority of its targets located within the sequences of genes, hundreds of base pairs away from the nearest 5' end of a gene. So again, not what you would expect on the basis of the Operon model. And I should stress that actually these transcription factor binding sites and promoters that are in unusual locations are actually perfectly normal binding sites and promoters. They've got perfectly normal sequences, that they function in the normal way, they bind tightly to their cognate protein in the test tube. The only reason we consider them unusual is because we're so biased in our way of thinking by the Operon model of genes and their control. There's actually a third paper that I'd like to point out down here. This is a paper from Bob Martin's lab at the NIH. Bob is now retired, but I think Bob was ahead of his time with this particular paper, and it was largely overlooked when it was first published. But what Bob's paper tells us is that actually you can take lots of coding sequences, okay, lots of sequences from within genes across the E. coli genome, and you find that many of those sequences actually have the ability to drive transcription, to drive the formation of an RNA. Okay, so completely unexpected. And it's these unusual transcriptional events that I'm going to focus on. I'm going to call the transcriptional events that I'm going to tell you about spurious transcriptional events, and I'm going to show you that they're particularly prevalent in parts of bacterial genomes that are AT-rich and have been acquired by horizontal gene transfer. So let me very quickly explain what I mean. Okay, so AT-rich groups of genes or operons clearly can be transcribed in the normal way. Okay, so you can clearly have a canonical promoter at the start and a terminator at the end, resulting in the formation of an mRNA. But what I'm going to show you is that many of these groups of AT-rich genes also very often are associated with the formation of spurious transcripts. Okay, so transcripts that initiate within genes, they might be in the sense orientation, they might be in the antisense orientation, but they're non-functional transcripts. And you're going to see that these transcripts arise in these regions because these regions of the chromosome are highly likely to contain spurious promoters, okay, so constitutive promoters within the sequences of genes. Very quickly, to finish off the introductory part of this talk, I need to tell you one more thing about AT-rich DNA, and that is that AT-rich DNA in the E. coli chromosome, okay, so I'm going to be talking to you about the E. coli chromosome, but much of what I'll say is actually applicable to many bacteria. AT-rich DNA tends to be bound by a DNA binding protein and DNA folding protein called HNS. The histone-like nucleoid structuring protein, okay? And HNS-like proteins are quite widely distributed in different bacteria. So in E. coli and closely related bacteria, you have HNS. In gram-positive bacteria, such as Bacillus subtilis, there's a protein called ROCK that seems to do the same job. In mycobacteria, there's a protein called LSR2, and so on. And these proteins bind to this AT-rich DNA, often in this extended filament or oligomerized format, as is shown on the slide. What does HNS do when it does this? Well, we've known for a long time that when HNS binds to these AT-rich sequences, it tends to silence transcription, but it's always been assumed that when HNS does this, it's silencing transcription from a genuine promoter upstream of these transcription units. And what I hope to convince you is that actually in many cases HNS is doing something much more complicated involving these spurious transcripts. So to quickly reiterate what I've already said, genome annotation is fine and can explain many of our observations, but it's heavily influenced by the Operon model and can't explain our most recent observations. So we need to start to study these hidden layers of complexity and even accept that they exist in bacterial chromosomes. Okay, so I'm going to focus on these AT-rich sections of DNA, and I've already told you that you're going to see that they're prone to these spurious transcriptional events. So why might this be the case? Why might these horizontally acquired AT-rich genes behave in this way? And actually, you can start to understand why they might behave in this way when you start to think about the DNA binding properties of RNA polymerase. So RNA polymerase, the enzyme that's responsible for the production of RNA molecules in bacteria, binds to promoter sequences, and bacterial housekeeping RNA polymerases tend to bind to two core DNA sequence elements called the -10 element and the -35 element. The -10 element has the sequence TA-TAAT, so an A or a T at all 6 positions. The -35 element, TTGACA, an A or a T at 4 out of 6 positions. And actually, if you align all known bacterial promoters, you see that it's the -10 element that's by far the best conserved. So actually, lots of promoters are perfectly able to function with a good -10 element and a relatively poor match to the -35 element. So RNA polymerase really likes to bind to and initiate transcription at these AT-rich DNA sequences. And this is just a consequence of these sequences being easy for the RNA polymerase to unwind in order to initiate transcription. But when we look at genes with an elevated AT content, we start to see why they're behaving in this unusual way. So what I want to do now is just show you 2 genes, okay, a gene with a normal AT content for the E. coli genome and a gene with an elevated AT content. And what you're going to see is that actually quite small increases in the AT content of the DNA can lead to disproportionate increases in the number of sequences within that gene that look like these promoter elements for RNA polymerase binding. So here's a gene called YHFK that I've just arbitrarily selected. I don't actually know what this gene does, but it's got an AT content of 48%, and this is fairly typical for the E. coli chromosome. You can find thousands of genes in E. coli with this sort of AT content. And if you look through YHFK for sequences that look like promoter minus 10 elements, you find that they actually occur very infrequently. So within YHFK, there are just 3 sequences that look like a promoter minus 10 element, 2 in the forward orientation, 1 in the reverse orientation. And I haven't just picked a gene here that fits the purposes of this talk. You can take any gene you like from the E. coli genome with this sort of AT content, and it will behave in a very similar way. Right, so now let's look at a horizontally acquired gene, an AT-rich gene. So we're going to increase the AT content from 48% to 61%, so just a 13% increase. This is a gene called the HXC. It's a horizontally acquired gene from E. coli 0157 that we worked on for a while. And if you look through the sequence of this particular gene, you find that it's absolutely infested with sequences that look like promoter minus 10 elements. So this relatively small increase in the AT content of the DNA, this 13% increase, has given rise to about a 10-fold increase in the number of sequences that look like promoter minus 10 elements. Okay, and what I want to do now is show you that actually many of these sequences that look like promoters are genuinely able to drive transcription of these spurious RNAs. So to try and convince you of this, I'm going to show you 3 lines of experimental evidence. I'm going to show you some biochemical experiments done in the test tube with purified DNA, purified proteins. I'm going to show you some genetic experiments, and then I'm going to show you some genomic experiments. So the first set of experiments involve the use of an in vitro transcription system. So we just have a purified DNA template. Okay, we add some purified RNA polymerase and some nucleotides, and then we look at the transcripts that RNA polymerase is able to produce. And our DNA template for these experiments is a plasmid, and this is a simple representation of the way that plasmid looks. And there are a couple of important things that I need to point out before I show you the data. So in this plasmid, we can clone a gene that we're interested in, represented by this grey arrow. And importantly, when we clone the gene of interest, which you'll see is going to be these horizontally acquired AT-rich genes, when we clone these genes, we just take the DNA from the start codon to the stop codon of that gene. So there is no way that gene can be transcribed in the normal sense. There's no promoter upstream. So any transcripts that are produced by cloning that sequence must originate within the sequence of the gene. The second thing to point out is that just downstream of the gene, there's a terminator. Okay, so any transcripts initiating within the gene will get terminated at that termination signal. And given the size of the genes that we've used in these experiments, between about a kilobase and a kilobase and a half, we expect to see these intragenic transcripts being a few hundreds, maybe 1,000 base pairs in length. So what do the data look like? So the first experiment we did was a control experiment. We used a normal gene with a normal AT content, not an AT-rich gene. And when we do that, this is the pattern of transcripts that are produced. And we get 2 sets of transcripts on the gel. So right at the top, you can see there are some transcripts that I've labeled plasmid. These are transcripts generated elsewhere on the plasmid. Obviously, the plasmid has got its own transcription units and genes that are transcribed in this experiment, and that gives rise to these large transcripts. Right down at the bottom of the gel, there's a transcript that's labeled RNA1. Okay, this is a small transcript. It's just over 100 nucleotides in length, and it's actually produced from the plasmid replication origin. And some of you will be familiar with the RNA1 transcript. When the plasmid's inside a cell, this transcript is important for controlling plasmid copy number, but in these experiments, it's just a useful internal control. But you can see the majority of the lane on the gel is completely blank. We don't see anything at all. So what happens when we put in horizontally acquired AT-rich genes? So I'm going to show you data for 4 different horizontally acquired AT-rich genes. They're all Y genes with complicated names, so I'm just going to refer to them for simplicity as genes A, B, C, and D. And when we put these genes into this system, you can see we get a very, very different result, okay? And in this region of the gel that was previously blank, we're now getting all of these different size transcripts, okay? And what we think is that these transcripts are due to these spurious promoters that are occurring within these genes because of their high AT content. And we can be sure that in this experiment that is the case, and that these promoters are due, these transcripts are due to internal promoters because we've made derivatives of each of these AT-rich genes where we've put point mutations in the internal promoters. And when we put point mutations in the internal promoters, you can see that most of these transcripts disappear. So in vitro, at least, on a naked DNA template with purified RNA polymerase, we can see that these AT-rich horizontally acquired genes are enriched for these spurious transcription initiation signals. So, of course, we next need to determine what happens inside the cell. Of course, it's much more complicated in the cell than in the test tube. So to try and answer the question as to what happens in vivo, we've generated the following scenario. So we've taken each of our genes and we've placed them upstream of a reporter. Okay, and again, when we've cloned the genes in these experiments, we've just taken the sequence from the start codon to the stop codon of the gene, so there's no upstream promoter DNA, and the only way that the reporter can be expressed is if there are promoters located within the sequence of the gene. Okay, I said it was more complicated in the cell, and of course one of the most pertinent complications is that these genes presumably are going to be bound by H&S. I told you earlier that H&S likes to bind to these AT-rich sections of DNA, and presumably when H&S does this, we're going to see that it blocks transcription from these spurious promoters. And that is indeed the case. So the tiny green bars that you can see there are data from wild-type cells when H&S is present, and you can see that we get very little reporter expression. So H&S is silencing transcription from these intragenic promoters. When H&S is deleted, you can see that the activity shoots up. And just a couple of points to make about these data. Actually, we can take lots of these horizontally acquired genes, put them into this system, along with their upstream DNA and their genuine promoter, and we find that actually they behave in an almost identical way. The data are almost indistinguishable. Okay, and what we think is that lots of genes that have been ascribed as being H&S regulated are possibly actually subject to this pseudo-regulatory effect due to these spurious promoters within these genes. Okay, the second thing I'd like to say is that in many of these cases, we can actually flip the horizontally acquired gene around in the other direction, and we still get very similar data, okay, because these genes are also full of spurious promoters in the antisense direction. Okay, can we mutate the internal promoters again and get rid of the effect? So we put the point mutations in, and when we put the point mutations in these promoters, you can see the activity comes right down, and this apparent regulatory effect that we refer to as pseudo-regulation now has actually disappeared. So hopefully you've found those two sets of data quite compelling, at least for these 4 AT-rich horizontally acquired genes. But of course, what we next needed to determine was whether or not this was a general phenomenon. Okay, so do all horizontally acquired AT-rich genes in the E. coli chromosome behave in this way? So to answer that question, we did some genome scale experiments, and we were very fortunate at this point to benefit from a collaboration with Joe Wade's lab at the Wadsworth Centre in Albany. So Joe and I were actually PhD students together, and we've stayed in touch since, so it's a very nice collaboration to have. And over the past 5 or 6 years or so, Joe has spent an incredible amount of time and effort in optimizing certain genome scale techniques, in particular derivatives of RNA-seq, to let him look at transcriptional events on a genome-wide scale. And we were able to apply some of these approaches to this problem. So essentially, in these experiments, what we did was map the distribution of as many promoters as we possibly could across the E. coli genome. And then we said, well, is there anything unusual about the positions of promoters with respect to coding and non-coding DNA when you look at parts of the genome that have been horizontally acquired and are AT-rich? So we identified in these experiments a total of 668 promoters distributed across the E. coli genome. And we identified these promoters using two complementary genomic approaches. The first approach that we used was a derivative of RNA-seq that Joe has developed in his lab. And using this approach, we were able to identify transcription start sites genome-wide at single nucleotide resolution. Okay, so for each one of these start sites, we can see exactly where the RNA polymerase is initiating transcription. We can see which direction transcription is occurring in, and so we can count back from that transcription start site and look to see if there's a promoter-like sequence further upstream. Okay, and we find that promoter-like sequences, minus 10 elements, minus 35 elements, are highly conserved upstream of these transcription start sites, which gives us a great deal of confidence that they're genuine sites of transcription initiation. So that generated one list of promoters. In a complementary set of experiments, we also used chromatin immunoprecipitation, or CHIP, coupled with sequencing, so CHIP-seq, to map the distribution of RNA polymerase on a genome-wide scale. So if you're not familiar with the approach, it doesn't matter. It's just a tool that we can use to measure protein-DNA interactions genome-wide, and we mapped the distribution of RNA polymerase. Importantly, when we did this, we only detected RNA polymerase. The way we did the experiment meant that we only detected RNA polymerases bound at promoters. So we didn't see any elongating RNA polymerase in the process of making a transcript. We just saw the RNA polymerase bound at the promoter, and that generated a second list of promoters. And we only included a promoter in this list of 668 if it was identified by both of those two approaches. So for every single one of these promoters, we can see the site of transcription initiation, we can count back and see a promoter-like sequence, and we can see that RNA polymerase is bound to that sequence. So we can be confident that these are genuine promoters. We then split this group of 668 promoters into two. So we had 160 promoters in H&S bound, 80 rich regions that were horizontally acquired, and we had 508 promoters elsewhere in the chromosome in the normal regions of DNA. And again, the way in which the data was split up into these two groups relied on the use of CHIP-seq data, this time CHIP-seq data for the binding of H&S across the chromosome. So numerous labs have mapped the distribution of H&S across the E. coli genome. The data are very consistent, and we're able to use that data to divide the promoters into these two groups. Again, we checked that the way we'd done this was reasonable, so we were able to measure the activity of these promoters in the presence and absence of H&S, and we find that the 160 promoters in this group here are generally suppressed by H&S, whereas the 508 promoters down here are generally not subject to regulation by H&S. So we have our two groups. We then divide them into four categories, each promoter into one of four categories. So we have promoters in intergenic regions correctly orientated to be driving the transcription of an mRNA. So this is the view of transcription consistent with Genome Annotation, the Operon model, and the textbook. We also found promoters in intergenic regions that were incorrectly orientated to be driving the transcription of a nearby gene or operon, and of course we found promoters within genes both in the sense orientation and in the antisense orientation. So how does the distribution of promoters between these four categories differ for our two groups, for the horizontally acquired genes and for the other genes in the genome? Well, if we look at the normal genes first, we find that they behave in largely the expected way. About 80% of the promoters that we detect in these parts of the genome are in intergenic regions, the two green segments. And of those promoters in intergenic regions, the great majority are in the first class, the class of promoters that would be suggested by the textbook. So pretty much what you would expect. Although I should point out that even in this scenario, we do find a lot of promoters perhaps not where you would expect them to be, but that's a story for a different day. Now, when you look at the AT-rich sections of the genome, these horizontally acquired regions, you find that you get a completely different result. And what you find is that actually now the promoters in the three unusual categories here actually make up the majority of the promoters that we see. Okay, so consistent with what we've seen in vitro with biochemical assays and using our genetic assays, we find that these AT-rich genes are full of promoters, and this applies on a genome-wide scale. And actually, we think that in this particular experiment, in the genome scale experiment, we're probably underestimating how often these spurious promoters occur. And we think that we're underestimating how often these promoters occur because, remember, the transcripts that are produced are going to be very unstable. They're non-coding transcripts of no apparent function, and they're going to be turned over very quickly by the cell, and that's going to make them difficult to find using techniques like RNA-seq. Okay, so we think that many of these promoters will have not made it into this analysis in the first place, and consistent with that, when we go back and do the genetics and the biochemistry, we find that these AT-rich genes are just completely infested. They're just plastered with these sequences that can drive transcription. So to give you an idea of how often we find these promoters for a typical gene of about a kilobase in length, we might find 10, 15, even 20 of active promoter sequences within those genes. So this would appear to be somewhat of an underestimation of how often this occurs. So to go back to a slide I showed you earlier, I told you that we'd known for a long time that H&S bound to these AT-rich genes and reduced transcription, but it was always assumed that H&S was doing this by silencing the transcription of an mRNA, silencing transcription according to the Operon model. And it's worth thinking about where this idea came from. And actually, very few H&S targeted genes have been studied in great detail using genetic and biochemical approaches. And when that has been done, it's tended to focus on what might happen to the genuine promoter located up here. And actually, this role of H&S in silencing canonical transcription has really been assigned on the basis of genome scale data, in particular data from the early microarray studies. So H&S is a very popular protein. Many people, some of them in this room, have worked on this system over the years. So of course, when DNA microarrays first became available, lots of people wanted to knock out the H&S gene and see what happened to the transcriptome. And of course, you see increased transcription of all of these horizontally acquired genes. But in retrospect, there were two problems with the early DNA microarrays. The first problem was that the microarrays tended to be designed on the basis of genome annotation. Okay, so you would never detect anything that you didn't expect to see on the basis of the way in which the genome had been annotated, which of course was biased by the Operon view of genes and their control. The second problem was that these microarrays tended to be fairly low resolution, so you'd perhaps have one or two probes on the array for each gene, and that made it almost impossible to differentiate between a genuine mRNA transcript and perhaps a transcript that initiated spuriously within the sequence of these genes. Of course, now we've got the benefit of more advanced genomic approaches, much higher resolution, and combining that with some focused genetics and biochemistry, we can see that what H&S is doing is actually much more complicated, and the majority of promoters that are silenced by H&S actually reside within these AT-rich DNA sequences. So why is this interesting? Well, my personal view is that this is interesting because we found a whole group of genes that behave in a completely unexpected way. It goes completely against the dogma suggested by the textbook, but actually there are other reasons to be interested in what I've told you so far. And many of those reasons are highlighted in papers. Sorry, let me just go back. Highlighted in papers previously published in this field of research, in particular related to horizontal gene transfer, the effects of AT-rich DNA, and the role of H&S. And what this huge body of very important literature tells us is that high AT content DNA actually imposes quite a major barrier to horizontal gene transfer, to the evolution of bacterial genomes by horizontal DNA transfer. And that's because these AT-rich sequences are actually quite toxic to the host cell. Okay, so when DNA comes into the bacterial cell that's more AT-rich than the host genome, that DNA is problematic for the cell. And we've known that for quite some time, and we've also known that H&S can counteract that toxicity. Okay, so it reduces the toxicity of this AT-rich DNA. But for me, when I've been reading these papers over the years, a missing piece of the jigsaw has always been an understanding of why this AT-rich DNA is so universally toxic to bacterial cells. And of course, what we now think is that it's something to do with these spurious transcriptional events. So I'm going to show you two more sets of data. I'm going to show you some data where we've taken some of these AT-rich genes, we've looked at their effect on cell fitness, and then we've looked to see if that effect is negated when we mutate these internal promoters. And then I'm going to show you one experiment where we've looked at the downstream consequences of spurious transcription for the cell. Okay, so we should be able to test whether or not the toxicity of some of these AT-rich genes is due to this spurious transcription by getting rid of these internal promoters. And you already saw earlier on in the talk that we can get rid of these internal promoters quite convincingly. So to do these experiments, we've returned to the plasmid that I mentioned earlier. So this is the plasmid we use for the in vitro transcription assay. And now we're going to put these plasmids into cells. We're going to put them into wild-type E. coli cells, and we're going to put them into a mutant lacking H&S. And I should say up front that we've double-checked that plasmid copy number is stable in all of these different genetic backgrounds, and it is. Okay, so we get about 65 copies of this plasmid per cell in both the wild-type cells and the cells lacking H&S. Okay, and what we're expecting is that when H&S is absent, we're going to see a growth defect. Okay, part of that growth defect is going to be due to the AT-rich DNA on the chromosome. Okay, but we're expecting the growth defect to be bigger than normal because we've supplied extra copies of these AT-rich genes. Okay, and we're going to look at what happens when you remove these internal promoters. So here's the results using the wild-type versions of these genes with their internal promoters intact. Okay, so we've got the same 4 genes, A, B, C, and D, as before. There's an additional gene here. Again, it's a Y gene of unknown function. I'm going to refer to that gene as gene E. In each one of those panels, you can see a growth curve. So we're looking at growth of wild-type E. coli cells, which in each case is the solid line, or the H&S mutant, which is the dashed line. And you can see that in each case, the H&S mutant grows more slowly than the wild-type cells. So what happens when we mutate the internal promoters that get rid of the production of these spurious transcripts? We find that the growth defect is reduced. Okay, so if you compare across between the different pairs of experiments, you can see that in each case, the growth defect is not as big when we mutate these internal promoters. So we know that these internal promoters are driving spurious transcriptional events. We can hypothesize that that might account for the toxic effects of these sequences, and that would certainly appear to be the case for the genes that we've tested so far. But of course, the question then is, well, why are these spurious transcriptional events toxic? And of course, it's unlikely to be the RNAs that are being produced that are poisoning the cell. The cell's full of RNA, it's perfectly happy to have all these RNA molecules floating around. And we think, quite logically, that what must happen is that in having to make these spurious transcripts, the cells are essentially wasting their resources. Okay, so you would expect this to deplete nucleotide pools. There should be various other consequences of having to make these transcripts. And even once the transcripts have been made, they then need to be removed from the cell, and that's going to be a further drain on resources. And we thought that the most likely and direct immediate consequence of having to make these transcripts would be a reduction in the pool of available RNA polymerase for all other genes. So to quickly explain what I mean using this hypothetical situation. So here we've got a housekeeping gene happily being transcribed by RNA polymerase and an AT-rich gene bound by HNS. And what I'm going to show you is that we think that when HNS is absent, RNA polymerase should get titrated away from this housekeeping gene. And to understand why that should be the case, I need to quickly tell you that the amount of RNA polymerase in an E. coli cell is limited. Okay, so there's only just enough RNA polymerase to go around all of the various housekeeping and basally transcribed genes. And there are two reasons for this. First, it would obviously make no sense for the cell to have more RNA polymerase than it needed. But second, having a short supply of RNA polymerase makes it easier for the cell to regulate transcription. Of course, if RNA polymerase was in excess, there'd be less of a requirement for activators, for example. So if there's a limited amount of RNA polymerase when HNS is absent and all of these spurious promoters are revealed, and remember we can find tens or even 20 or so of these promoters within these horizontally acquired genes, and there are hundreds of such genes across the genome, we'd expect these promoters to start to compete for binding RNA polymerase, and that should reduce the amount of RNA polymerase available for housekeeping genes. So this model should be quite easy to test. Okay, so you should be able to do a transcriptomics type experiment, knock out HNS, and you should see that transcription of all basally acquired genes, anything that's been transcribed under normal growth conditions, should be downregulated in an HNS mutant. And not only should all of these genes be downregulated, but they should be downregulated to a very similar extent. There should be a uniform downshift in all basal transcription. Okay, and that should happen because the pool of available RNA polymerase has got smaller. And as I've already mentioned, lots of people have used transcriptomics to study an HNS mutant. But actually, if you look in the literature, there's absolutely no evidence that this takes place. So we started to think why that might be. And we came to the conclusion that it was probably a consequence of the way in which transcriptomics type experiments have typically been normalized. Okay, so just to quickly tell you what the problem might be. So when you do a transcriptomics experiment, you'd extract RNA from wild type cells, you'd extract RNA from a mutant, in this case an HNS mutant, you'd then probably reverse transcribe that RNA and then label the sample. Okay, so you'd have two samples of a reverse transcribed DNA ready to, in these many cases, put on a DNA microarray. Now, the problem is that the RNA extraction, the reverse transcription, the labeling all occur with varying efficiency. Okay, so by the time you get ready to actually analyze in your sample, they're going to have been processed quite differently, and that can lead to technical artifacts in the experiment. So when you actually look at ratio changes in gene expression, you actually find that it's very difficult to find genes where transcription doesn't appear to have changed because of these differences in the way that the two proteins, the two samples have been processed. Okay, so it's very difficult to find genes where the expression ratio is 1-to-1. Okay, so to get rid of those problems, the data are normalized. Okay, so the 1-to-1 ratio is artificially set across the genome on the assumption that the transcription of most basally transcribed and housekeeping genes hasn't changed. But what we're arguing is that actually the transcription of these genes will have changed, and that by applying these normalization procedures, actually the effect that we're expecting to see will have been removed. So there are now various ways around this by using internal RNA standards. For example, we've developed a different way of doing it. I'm not going to go into the details, but it allows us to see what we expected to see. And we indeed see that there's a blanket, very uniform downshift in all basal transcription when HNS is deleted. So I'll just show you two more data slides. So here's a small region of the genome, similarly organized to the cartoon on the last slide. So here we've got a very well-defined HNS targeted locus, the pro-U locus, and you can see that when HNS is deleted, we see increased transcription in this region. Just over here, we've got some important housekeeping genes involved in the production of tRNAs, and you can see that they're clearly down-regulated. I use the word regulated probably inappropriately, but the level of transcription decreases. And what we see is that this decrease in basal transcription is uniform across the genome. So here's the data for the whole chromosome. So each one of these data points represents a gene somewhere on the E. coli chromosome. The 80 rich H and S-bound genes are shown in red. All of the other genes are shown in black. And the pale blue line that you can see here represents where each of these data points should fall if there's no change in transcription between wild-type cells and cells lacking H and S. And what you can see is that actually very few data points fall on this line. And just to make the point again, in many transcriptomics experiments, the data would be artificially shifted so that many data points did fall on this line. And what we actually see is that we obviously get the increased transcription of the H and S-bound genes, but if you look at all of the other genes, the black data points, you can see that they behave in a very uniform way, and transcription is downshifted. Okay, so that's the majority of what I want to say. I'll just quickly sum up so I started off by talking to you about the operon model of transcription. I told you that it could explain many of our observations, but it could no longer explain everything, and that we've become somewhat biased in our thinking to assuming that this is the only way that genes can behave. And what I've shown you is that in order to understand horizontally acquired AT-rich genes, and particularly their toxic effects on cells and their unusual properties, we need to go beyond the operon model and understand the hidden complexities of these genes. So just to quickly thank a few people. So here are some pictures of Birmingham in the sunshine. Birmingham does actually look very nice in the sunshine if you've ever been and the sun has shone. These are the people in the lab, of course the most important people. So most of the work that I've talked to you today has been done by a combination of Shivani Singh and Lisa Lamberti. So Shivani was a PhD student and now a postdoc in the lab. Lisa has just finished writing her thesis. She handed it in last Friday, and Lisa did most of the more recent experiments that I talked to you about. And this work has been funded by a combination of the Wellcome Trust and the Leverhulme Trust. And thank you very much for your attention.